Good afternoon, everybody. My name is Chijioke Nze, and this summer I had the pleasure of working with Dr. Nate Dudley in the Rothman lab. And our project is focused on understanding the molecular mechanism of totipotency using C. elegans. Before testing in humans, we do testing in animals as a reference before going to do testing in humans. This is made possible because of the common descent of all animals, as well as the conservation of metabolic developmental patterns and genetic material. We use C. elegans as our model because they're relatively small, about 1 millimeter long. They can go from embryos to reproductive adults in about 3 days, so they have a short generation time. We know their developmental patterns, so we can track any changes due to genetic manipulation by us. They're transparent, so we can see their internal organs, and their entire genome is sequenced, so we know what goes in and what comes out. Early in embryogenesis, around just a few cells, all the cells in an animal are totipotent. Totipotency is the potential of a cell to differentiate into any cell of all 3 germ layers when given the appropriate signals. As time goes on, the cells begin to lose their potency or their plasticity and become more destined for a particular cell type till they are restricted in potential. This window of plasticity is what we call the competency window. I don't know how many of you guys are familiar with the show Heroes, but this is Claire Bennett. Claire has the ability of regeneration. Regeneration is a combination of rapid cell division as well as appropriate cell differentiation, which requires totipotency. Our project is focused on the totipotency aspect. In the long run, we hope to identify and characterize all the genes required for maintaining totipotency in the hopes of maintaining a stem cell-like state even late in development. This project has numerous applications in injury and disease, including tissue loss, many cancers, multiple sclerosis, Alzheimer's, the list goes on. My project this summer, however, was to understand the role of a particular gene, Humpty, in maintaining totipotency. A previous study done in my lab showed that the knocking out or suppression of this gene caused a reduction in the span of competency. Because of this, we hypothesized that overexpressing this gene could potentially increase the span of this competency and hopefully even increase competency late in the window. Once again, competency is the ability to respond to signals that we give it. Now, before we begin testing, we need to establish a control as a reference so we can know how our genetic manipulation affects it. And to do this, we culture worms, and then we build a competency window. How do we build a competency window? Well, the strain of worms we use has a couple of constructs inserted into its genome, one of them being heat shock promoter attached to an N3 determinant. N3 determinant is a gut of signal that tells the cells be gut. So this means whenever we heat shock the animals, this construct becomes activated and tells all the cell in the animal, be gut, be gut, be gut. We track the responsiveness to these signals using our second construct, L2 GFP construct. Now L2 is a gut-specific marker present in all and only gut cells, and GFP is green fluorescence protein. So all gut cells have L2 and therefore will fluoresce green. As you can see, this is a C. elegans with gut cells, the nuclei of the gut cells glowing green. And we give them the signal with this, and then we track how many of them are transformed. And that's how we build our competency window. Now to go back one slide. This shows the competency window previously described in our lab. As you can see, this actually shows the percent transformation, which shows responsiveness or competency at these cell stages. So as you can see, the peak is around 22 to 42 cells. That's when they're most responsive to our signals. Now this is what a transformed cell looks like. As you can tell, these right here are big balls of gut cells, which means we got the entire animal, skin cells, hair cells, eyeball cells, everything turned into gut cells. And so we'd score these as transformed embryos. However, these animals have only about 20 gut nuclei, probably lining the digestive tract of the animal. So these would be untransformed embryos. And we use this to build our competency window by measuring the percentage of transformation versus non-transformation. After establishing a control, then we test the effects of our genetic manipulation on the competency. For one, we use RNAi, which is a gene. Well, RNAi is a highly conserved gene silencing phenomena, which directs the sequence-specific degradation of complementary mRNA. This means that we can suppress genes by destroying the mRNA before translation of a particular gene we want, if we know the codon for the complementary pair. So using this, we knocked out our gene, Humpty, and some other related genes. Then we score the effects of knocking out Humpty on the competency by building another competency window using animals that we've gotten to not express Humpty. And we compare that to our control, which is where the control comes back in. After we've established the role of Humpty on the competency window, then we build a transgenic worm which overexpresses Humpty by inserting another construct into our animals. And then we build another competency window so we can know how overexpression affects the competency. And then we compare the new window with the control window, and this tells us how it affects it. Now this is the control graph that we built. If you notice, it looks a little different from the one I showed before with only one peak which goes up and goes down. We actually noticed an interesting trend. It seemed to be kind of bimodal with peaks around 22 to 40 cells and 100 cells. Now this was interesting, kind of threw me off a little bit, but this might be because this is the first time this window has been so extensively defined. Usually a lot of the later endpoints are put in a clump, so you don't really get the real feel for what the competency window looks like. Next, this is the results of the RNAi knockout of our gene Humpty. We noticed, however, that in the early part of the window, as you can see, there seem to be two regions, early and late window of competency with two peaks. We are only able to define the first region of competency. As you can see here, it looked like there wasn't much difference between our control and our knockout in the early region of the window. This was sort of surprising because we've seen these results before. However, we haven't defined the later part of the window. So we believe that this part of the window might be controlled by another gene. And in addition, the initial experiment that led to this project was actually done later in the window where the suppression of the competency was done. So we believe there's still hope for this region of the window. It's a big gap. So we concluded that the competency window of wild-type worms appears to be bimodal with peaks of 20 to 40 cells and 100 cells, and that Humpty does not influence competency early in the window. Competency in this period might be regulated by another gene, which we have an idea of what this gene might be, but no testing has been done yet until we conclude the second part. Our future directions, we want to define Humpty and other related genes' effects on competency late and early in the window. We want to create a construct that overexpresses Humpty, and then we want to create a worm that overexpresses Humpty by inserting this construct into the worm. And then we want to define the competency window of the transgenic worm that overexpresses Humpty and score the effects late in development. I'd like to thank my professor, Joe Rothman, for allowing me to work in his lab, and the amazing people at the Rothman Lab for all their food and support. I'd like to thank my amazing mentor, Dr. Nate Dudley, for teaching me the ropes, and also my semi-partner. I'd like to thank UC Leeds for funding me. I'd like to thank Eric and Math for their patience and guidance through this process, CNSI, and you guys. Thank you. Questions?